Hi, everyone. Welcome to the Physionic podcast. My name is Nicholas Verhoeven. I'm a PhD candidate in molecular medicine. And today I'm going to attempt to discuss yet another pretty involved paper, but this one is actually going to be, I think, pretty useful to people that suffer from high blood pressure. Mainly because this study that I'm going to be going through discusses high blood pressure, and they come up with a mechanism for why high blood pressure occurs. So as usual on Physionic, I'm going to be going from a really high level to try to introduce this story, and as we go through, we'll be getting more and more into the weeds, and then at the end, I'll have a conclusion and some of the takeaways, certain things that we might actually be able to do to mitigate high blood pressure. And it's actually not things like reduce your salt intake or reduce your calories, which obviously are great recommendations, but this is far more targeted to people that potentially have this problem. So with that said, if you're watching the podcast, of course, I'm going to have a bunch of graphics that are going to pop up. Some of it is going to get kind of molecular. I try to keep things as kind of visually appealing, and of course, I'll be explaining things for anybody that's just listening. And also, I'm skipping maybe 80% of the data. So I'm just highlighting the main points, and so that we can tell the story, but I will have the paper linked for you so that you can look at the data for yourself if you're so inclined. Okay, so let's begin. Hypertension or high blood pressure, those are one and the same, is caused by, or one of the presumed ideas as to what causes it is peripheral resistance. So you have your heart that's pumping, and as it's pumping, there's resistance across all your arteries, across all your veins, but more specifically across your arteries. And that resistance that gets caused can be due to a variety of different things. I created a video on this, I don't know, maybe 4 years ago. So you can have kind of turbulent flow of blood. So as the heart is squeezing, obviously you have blood flow that's moving from the heart across into the arteries. And you can have kind of a disjuncted, a non-flowing version of that blood flow where it's turbulent and it kind of hits the walls and it pushes up against the walls, and the walls of the arteries have to, because they're kind of like tubes, but they're dynamic tubes, they squeeze in and they try to resist that flow. So that is one of the causes of this peripheral resistance, peripheral being outside of essentially the heart in this situation, potentially the aorta as well. So when you have greater levels of this peripheral resistance, then you tend to have higher blood pressure. Now, these researchers wanted to look a little bit more on how these vessels, how these tubes, how these arteries, those are all three the same thing, how they react to pressure that's being pushed onto them. So if you think of, let's say you've got an artery. As a matter of fact, let me throw up this first graphic here for anybody that's watching. When you think of your entire circulatory system, your entire vasculature, I'm talking your arteries, I'm talking your veins, your venules, your arterioles, not as much with the capillaries, but certainly some of this still applies. But ultimately, the entire tubular network that emanates from your heart and goes to your lungs and goes to your muscles and goes to your brain, all of that entire circulatory system is essentially just a bunch of tubes, as I mentioned, but those tubes have the ability to constrict, known as vasoconstriction, or they have the ability to widen. So you think of like a hose. I always use that as my great example. And with that hose, the hose can enlarge to reduce blood pressure, or it can constrict and therefore increase blood pressure. So in the bloodstream, obviously we've got red blood cells, and we've got a bunch of other like minerals and different other cells. You would have like monocytes are an example. So you've got immune cells in there. So you've got a bunch of stuff that's going through that. And of course that takes space. So if you have a limited amount of space, then it's going to create pressure. So one of the issues with the constriction of these vessels is that, well, you're decreasing the amount of space that's existent. So with that, then you're going to have greater pressure on the overall tubular network, and that's where we get this high blood pressure. So how does that happen? How does that actual constriction occur? And that constriction occurs due to smooth muscle cells. So smooth muscle cells are not on the liquid side of this tube. So this tube is, let's say, thick, and the insulation of that tube, so if you were to look straight down into a hose, you can see the hole. That's where all the liquid is coming out. And then you've got the insulation that actually makes the hose. And part of that insulation in our tubular network, in our circulatory system, is made up of smooth muscle cells. Now, why would muscle cells be there? Well, muscle cells are there because those are the things that are going to end up constricting the tube. And you might be thinking, okay, well, let me just get rid of those, right? No, you don't want to do that either, because you do need blood pressure. You need a certain amount of blood pressure. Otherwise, you end up dying from a lack of blood pressure. So we need to be in this sweet spot. But for some people, for a lot of people, as a matter of fact, they suffer from high blood pressure. So, and the idea here by these researchers is that there's an overconstriction of that tubular network, as the smooth muscles are undergoing what's known as myogenic tone. Okay, so that myogenic tone is what allows for that constriction of the tubes, and therefore an increase in blood pressure. So this increases peripheral vasculature, or peripheral vascular resistance. And so if you have this increase in this pressure in the periphery, then obviously if you measure it at the arm, or if you measure it at the ankle or the calf or whatever, it doesn't matter, you're ultimately going to get a reading that's a high systolic blood pressure and a potentially high diastolic blood pressure. But usually people focus, or clinicians in general focus more on the systolic blood pressure, because that's the highest level of blood pressure that the circulatory system is experiencing at any given time. So in previous papers, they've looked at mechanosensation. You can imagine, as I've just been talking about, that there's an increase in pressure. So that pressure obviously affects all of the cells that make up the tube. So if they sense the pressure, then they can react to that pressure. And while that's incredibly important, maybe even the most important, it's still not everything. And these researchers wanted to know, although other papers have investigated mechanosensation, the ability to sense this pressure that increases myogenic tone again being the constriction through the smooth muscle cells, are there potentially other mechanisms like extracellular chemical signals? What I mean by chemical signals is like different proteins, different cytokines that could potentially modulate this effect as well. So that's ultimately what they want to do. They want to use this model that they think might be working and leads to this increase in blood pressure. So just to round things out one more time, you've got peripheral resistance that occurs on the inside of this tubular network. That's our circulatory system. Our circulatory system reacts to that. The cells that make up the actual tubes themselves, the smooth muscle cells, will constrict. They will close that hole that the liquid can go through, and suddenly blood pressure increases. So the signal that they wanted to look at was a particular one. They wanted to look at TGF-beta, which is one that I've discussed in actually relatively recently as well. And this TGF-beta is a cytokine. It's a communication molecule that gets used for all kinds of different things. TGF standing for transforming growth factor. And ultimately, how these growth factors are produced is through an immature form of the protein. And what happens is that immature form gets cleaved, meaning that it gets cut. And it gets cut in such a way that it leads to the production of the fully mature TGF-beta. And that fully mature TGF-beta can then bind the cells and lead to this change. And in this case, we're looking at smooth muscle cells. So what the researchers wanted to know is how another protein known as Emilin has an effect on TGF-beta. So canonically or traditionally, Emilin will bind the immature form of TGF-beta, meaning a pro version of TGF-beta. So just know I'm just going to call it the immature form of TGF-beta. And that is a form of TGF-beta that still has its immature pro subunit, subsection attached to it. And the only way that it can become active is if that pro section gets cut off, and then you get a fully functioning TGF-beta molecule that can then affect the cells. And we'll see how TGF-beta affects the cell specifically in just a little bit. But what Emilin does is it actually binds this immature form of TGF-beta, and it stops the cleavage, the cutting off of that pro section. Therefore, the TGF-beta levels decrease. So the circulating TGF-beta levels then decrease as a result. So with that in mind, if you have lower levels of Emilin, the idea would be then you would have increases in TGF-beta, not because you're producing more necessarily, but because you've got less that's sequestered and bound to Emilin. So Emilin is like, it's just sequestering all this TGF-beta. And in a previous podcast, as a matter of fact, just a few weeks ago, I talked about another protein that sequesters TGF-beta, which was fibrillin. So now we've got this particular protein, Emilin. So that's what we're going to be focused on. And the researchers they're going to be using a lot of mouse work, but at the end, they are going to be relating this to humans in clinical situations. So that's what makes this paper ultimately all that translational and really interesting. Okay, so that is kind of the introduction to understanding how this all works. And I'll of course be explaining how TGF-beta specifically, and you'll see this if you're watching the podcast, you'll see the effect that TGF-beta has on smooth muscle cells. Again, just one last time, an increase in myogenic tone means that the smooth muscle cells are contracting, and if that is occurring, then you get an increase in blood pressure. That's the idea. Okay. Now, let's dive into our first piece of data. And the first piece of data, these researchers essentially just wanted to measure blood pressure. So what they did is they literally genetically modified mice to express this Emilin gene, but on either side of the Emilin gene, they used a particular gene sequence. I'm being very general here, but they have two gene sequences or gene targets that are on either side of the Emilin gene, and which would allow the mice to actually produce Emilin if the Emilin gene is present. And once the mice are fed a particular substance, that substance triggers the cells, the smooth muscle cells, to cut out the Emilin gene. So therefore, the mice have the Emilin gene for a while, and then once they're fed this particular substance, in this situation it's tamoxifen, that leads that gene to get cut out. And obviously you can imagine now they don't have the Emilin gene. So this is a really cool model and used a lot when it comes to molecular biology and physiology and et cetera, et cetera. So really simple experiment. All they wanted to know is if Emilin is present and what happens when Emilin is gone. And Emilin, again, is that protein that sequesters TGF, and it's outside of the smooth muscle cells. So it's in what's called the extracellular space. And what happened is that for a few days, even with this removal of this Emilin gene, there seemed to be no effect. So for about 6 days, 5-6 days, there's about no effect. And then suddenly there's a rise in blood pressure, a rather significant rise in blood pressure that's systolic and diastolic. And if you're looking at the data, the systolic is up top and the diastolic is below. And it's a pretty significant rise. I mean, good, I don't know, say like 15 to 20 millimeters of mercury. That's a huge increase in blood pressure. So here we've got proof that the removal of Emilin leads to an increase in blood pressure. Okay. Now, they wanted to take it a step further, and they're going to be taking it many steps further. But then they wanted to know, okay, so this is just in whole mice. Now we want to see what happens when we remove particular arteries. So they actually removed arteries from the mice, and then they did the tests on the arteries themselves. And they found they're measuring myogenic tone. Again, smooth muscle cells contracting, leading to this constriction of the arteries, which would presumably be related to an increase in blood pressure. And they did the exact same experiments, again, with tamoxifen and removing Emilin or keeping Emilin in place. And they found that only in one condition did myogenic tone increase rather substantially by about 12%, something like that. And that was with the Emilin absent mice. So if Emilin wasn't there, then you saw an increase in myogenic tone. So this feeds into the story. It continues our story going forward. So at this point, we know that Emilin is implicated in some way to this increase in blood pressure. So the takeaway from here is that the loss of Emilin leads to greater myogenic tone and blood pressure. Okay? They also did the same thing with TGF-beta. So just ignoring Emilin, they just wanted to see, okay, if we just add TGF-beta to the arteries, just squirt it on there, or just have, let's say, the arteries may have been on a plate or something like that, may have been suspended in some sort of liquid. I'm not entirely certain on that. But ultimately, they surround it in more TGF-beta than it's usually around. And although I'm not going to show the data, there was, again, the same effect, an increase in myogenic tone. So now we've got Emilin and we've got TGF-beta that are increasing myogenic tone and therefore potentially blood pressure. So the next step that they wanted to go into was to look at calcium measures. Now, why would they look at calcium? You might be asking, where does calcium roll into this? Well, calcium is the most important molecule, arguably the most important molecule when it comes to muscle contraction. Certainly true in skeletal muscle. It's also true in smooth muscles, just through a different mechanism. But ultimately, it leads to the contraction of the musculature. So if you have an increase in intracellular, meaning inside the cell, inside the smooth muscle cell, you have an increase in that blood pressure, then that means that you would typically have an increase in the contraction. So myogenic tone. So that's why they wanted to look at calcium. But they didn't want to just look at calcium. I'm going to throw up the data real quick, but they didn't want to just look at calcium. They also wanted to use an inhibitor for a particular calcium channel. So now they're getting at kind of the mechanism as to how this is occurring. So let me cover the calcium first. So again, they're taking arteries from animals that have Emilin and animals that do not have Emilin. Emilin being the protein that sequesters TGF-beta. And they saw that when Emilin was absent, so was not in the arteries, there was an increase in the amount of calcium that was influxing, that was going into the smooth muscle cells. So again, this kind of shows more proof that you've got greater contraction, greater myogenic tone. Now, to see, okay, what would happen if we use a drug that inhibits a particular channel? What I mean by a channel is not a TV channel. I'm talking about a protein that's embedded in the cell membrane, and it allows calcium in. If you guys promise not to freak out if you're watching the podcast, I'm going to throw up a graphic real quick, and I'm going to go back to the data. And it's busy. I get that. I'm going to explain everything as we go through, but of course I'm going to explain it again at the very end so that we can all piece this together. But you can see there on the far right, and if you're listening, don't worry, I'm explaining it, that there are these pores that are made out of proteins that are embedded into the cell membrane, and that allows calcium to enter the cell. So a particular protein or particular channel that they're interested in is TRPC channels. And so TRP, but they're kind of said as trip channels. So we're going to go back to that in just a little bit. But the point is that they use an inhibitor for that trip channel. And they found that it got rid of the effect of increased calcium into the smooth muscle cells in the Emilin absent mice. So the mice that did not express Emilin. So from here, then they wanted to know, okay, so we know that that's the case in these mice. What happens if we use just regular arteries and we end up adding TGF-beta? And they saw the exact same effect. They saw that calcium increased to TGF-beta, but it was decreased. The effect was massively decreased when they added this particular inhibitor that inhibited this trip channel. So now calcium is not entering as much. Now, clearly there's still some calcium entering, but it's not more than the standard condition, the vehicle condition. So a condition where you're not adding TGF-beta or you don't have Emilin absent. So this essentially says that calcium influx is mediated by these trip channels. Okay, so that's important as we go on. So calcium influx into the smooth muscle cell, which leads to increased contraction, which leads to greater myogenic tone, which leads to blood pressure, increases in blood pressure, is mediated by these trip channels. And TGF-beta increases the influx of calcium, and Emilin deficiency also increases the influx of calcium. Okay, now they want to focus in more on TGF-beta itself. How is TGF-beta doing this? They're just throwing it on there and they're somehow seeing these effects. What's going on? So they end up looking at downstream signaling, meaning that they add TGF-beta. I'm going to throw up my graphic real quick. They throw on TGF-beta. TGF-beta binds its receptor on the cell membrane, and then there's a bunch of things that happen that I did not depict. And then you get the gene expression of particular molecules, particular proteins. And that's where I'll stop that explanation because we're about to touch on it with the actual data itself. So they did the exact same experiment. Here in this situation, they're looking at 3 different genes. So they're measuring ADAM17, EGF, and EGFR. Now let me explain those. So ADAM17 is a protein that when EGF is produced, it goes to the cell membrane. Let me go back to my graphic here. So when the gene is read and the protein is produced, it is sent up to the cell membrane and it kind of just sits in the cell membrane. So there's a piece of the protein that's stuck in the cell membrane. Now what ADAM does is it sounds kind of funny, Adam, like a person. Jim and Bob are also involved. No, it's just Adam comes in and cleaves that protein part that's stuck in the membrane. That now releases EGF from the cell to then bind its EGF receptor. Okay, that's where I'm going to leave that. So just know that Adam plays a factor in cleaving EGF and allowing EGF to get released. So now looking at how that is overall going to be affected. So then they look at EGF, the actual production of EGF itself. So not only the production of ADAM, that cleavage, that protease that cleaves EGF at the cell membrane, but they also look at EGF and they also looked at the EGF receptor, EGFR. Okay, so with that all said, when they didn't add anything to the cells, or more specifically to the arteries in this situation, they found no difference, which obviously that makes a ton of sense. If you're not going to do anything to the arteries, of course nothing's going to happen. Well, at least you would hope. Then they added TGF-beta like they did in their previous experiments, and they found that one gene was highly expressed, and that gene was EGF. So not EGFR, not the receptor, and not ADAM17 that cleaves EGF, but a substantial increase in EGF. And then they put TGF-beta back onto the arteries, as well as a TGF receptor inhibitor. Because remember, TGF cannot actually act any of this out on the actual cells themselves unless it binds its receptor. So, as such, that means that you are annihilating or getting rid of TGF-beta's activity or its ability to function. So, as such, then that means that you would presume that everything downstream of that TGF-beta receptor would be gone. It would be just like when you didn't add TGF-beta to these cells or these arteries. So, what happens? Well, they find that if you're looking at the data, that it's exactly the same as if they didn't add TGF. So this is a huge implication here. They're definitely showing that EGF is upregulated when TGF, I keep throwing those around that I hope I'm not getting too confusing. TGF is the one that we're most focused on, but EGF is the product. So when TGF is added to the arteries, we see this increase in EGF. And then when we block TGF, then we see EGF go back down. Okay, so that was pretty telling. Then they want to look at the smooth muscle contraction, the myogenic tone, in the exact same kind of scenario where they add TGF, or they add TGF plus an antibody for EGF. So now they're eliminating EGF, but they're still keeping TGF. And they find that that actually leads to an inhibition of myogenic tone. So when TGF-beta, just like in all their other data, showed an increase in myogenic tone when they block EGF, so that's downstream from TGF receptor, then that meant that you saw a removal of that myogenic tone. Ooh, I should have come up with like better names than TGF and EGF, because I feel like I'm continuously saying them back and forth, and hopefully you're able to follow along. That's tough. Okay, so what does that mean? What does any of that mean? This tells us that TGF, transforming growth factor, beta, increases EGF. I forgot what that stands for. Something growth factor. Yeah, it's epidermal growth factor. No, it's definitely not that. I can't remember it off the top of my head. But EGF is again downstream of TGF, transforming growth factor. So this tells us that TGF, transforming growth factor beta, increases EGF expression, and inhibiting EGF stops this increase in myogenic tone. So once EGF is cleaved and it leaves the cell, if you bind it, if you sequester it before it can come back and bind the receptor, the EGF receptor, then you get rid of myogenic tone. So clearly there's a mechanism there that's starting to get elucidated, that's running through TGF-beta, that's running through Emilin, but we're focused on TGF-beta at the moment. Okay, so now that was a lot of information. That was all in mice. Now we're going to be focused on what happens in humans. Do we see any of this stuff in humans? Because if we don't see any of this stuff in humans, it's kind of like, cool story, bro. It doesn't matter. So they were interested in this, which I'm glad that they were, because it makes things a greater story as a whole. Okay, so now they do the exact same experiments in humans where they had patients that were going through hernia surgery. And all they did was they tested their blood pressure. So they just checked, hey, do these people have high blood pressure? And that's it. That's all they did. They just wanted to check, and they found that a certain population of individuals that went for hernia surgeries had high blood pressure, and a certain population had normal blood pressure. So great. Now we've got our two conditions. We've got our difference there. High blood pressure, hypertensive individuals, and normal-tensive individuals. And they took some fat samples from these individuals, and then they removed some arteries from those fat samples because, well, people probably don't think about it, but fat has to be fed blood as well. So they were able to extract arteries from the fat tissue. And obviously, these people aren't going to be missing their fat tissue. Well, not at least a small amount. So as they were going through their hernia surgery, the researchers got some fat tissue, then from the fat tissue removed some arteries and did the exact same experiments. So the first test they did was on myogenic tone, and they found, shockingly enough, the high blood pressure individuals had elevated myogenic tone compared to the normotensive, the normal blood pressure individuals. Okay, so that corroborates what we've been talking about so far in mice. Now the next thing was to, all right, how about we inhibit through TGF-beta? How about we inhibit TGF-beta and see what happens? And it turns out that when they plate these arteries and they add this antibody, this specific molecule that binds TGF-beta, then you see a reduction back to normal blood pressure. So here we have evidence that if we inhibit TGF-beta, then that means we get a removal of that high blood pressure back down to normal blood pressure. But that's not the end of the story. So still, that's really, really interesting. And it tells us that the mechanism that we're looking at is definitely in humans as well. Okay, and the final piece of data before we get to the conclusions, kind of pulling all this together, and some of the takeaways as well, is then they did the exact same thing. So they took those arteries and then they cut them so that they could see kind of a histological view of these arteries. And then they stained them, so they used molecules that are specific to Emilin. And they just wanted to know, the only thing they wanted to know is between the hypertensive individuals, the high blood pressure individuals, arteries from them, as well as arteries from the normal individuals with normal blood pressure, if there is a difference in Emilin amount. And what do they find? So if you see greater expression of that purple expression, if you're watching the podcast, then that means there's more Emilin. And what they found is in the normotensive individuals, the people with normal blood pressure, they had much higher levels of Emilin. So now this starts to kind of bring in this whole story of Emilin and TGF-beta and how this is all playing a role. Okay, so what's the conclusion from all this, and how do we fit all of these mechanisms together to create a concrete story out of all this? So having low levels of Emilin leads to raised TGF-beta levels. Let me go ahead and throw my graphic up here. So TGF-beta is normally sequestered by Emilin. And when it's sequestered, it can't interact with the smooth muscle cells. But if you have low Emilin levels, suddenly you've got less of this protein that's sequestering this TGF-beta. So TGF-beta is then being activated, and it's binding this TGF receptor. Now that TGF receptor leads to a bunch of signaling inside the cell that I did not go over at all, because they actually didn't go over it either. And that then leads to much greater EGF gene expression, leading to the production of more of this EGF protein. The EGF protein then gets pushed up to the smooth muscle cell membrane, and it sticks there. Then the ATOM protein will cut it, and I do not show this in the graphic because it was already overwhelming and I really didn't want to scare people off. So that ATOM protein, that protease, will cut the EGF protein off of the cell membrane. The EGF will then be free, and go into the extracellular matrix, into the extracellular space outside of the smooth muscle, and it will bind either other smooth muscle cells, or it will go back and bind itself, and which is known as an autocrine action. And the EGF molecule protein will then bind the EGF receptor, which will lead to a series of other signals within the cell, kind of like the TGF receptor had its own other signals. That then triggers the TRP channels that we were talking about earlier, leading to an influx of calcium from outside of the cell into the cell. Then, not shown and something that they also looked at, was another channel that actually activates another channel, And then that activates yet another channel, which is a much bigger calcium channel known as a voltage-dependent calcium channel. So you have a small amount of calcium that comes in once EGF binds its EGF receptor from the TRP channel, and then that indirectly ultimately leads to a much greater influx of calcium through this voltage-gated calcium channel. That then leads to muscle contraction and therefore myogenic tone. Wow. Okay. So this is all through these TRP channels, through this EGFR, through the TGF beta and the lack of Emilin. And we see that there's obviously an association. At least we can retrospectively say that if we do some of these measures in humans without any manipulations, we just test their myogenic tone, we just test their blood pressure, we just test the amount of Emilin that's present, and we find consistently that it's true for those, just like we did with the more causative data from the mouse models. Now, do we need more information? Yeah, of course. But this is still pretty telling information. So this is also ultimately this is found in humans. And if we look at the human samples with high blood pressure, at least, so the mechanism likely acts in humans just as it does in the mouse model that we talked about. So what can you do about this? That's probably something that people want to know about. At this point, I probably don't have to tell you. You probably have an idea of what are some things you might want to look into. It doesn't have to be super specific. We're talking about TGF beta. What does TGF beta do? It increases myogenic tone. So maybe trying to decrease the levels of TGF beta. That would be something that a clinician could do through some sort of drug. Obviously, there are problems with that because there's all kinds of side effects that are going to come from drugs. Some other more, I guess, quote unquote, natural ways of decreasing TGF beta. You may want to look some of those up and see if you can decrease your TGF beta. Another one is maybe forget TGF beta and you want to go downstream of TGF beta and you want to look at EGF. That could be another option, trying to decrease your EGF levels. And finally, increasing Emilin amount. Now, if that is a genetic issue, that could be problematic. Then in that case, you'd probably need some sort of genetic treatment, which a lot of countries don't allow at the moment. Or if we eventually find a way to increase Emilin levels, then that could increase the levels in our arteries, and then that would then lead to this decrease in myogenic tone. So really focusing in on any one of these pathways, and another one that people might say is, well, let me inhibit calcium. Unfortunately, calcium is so ubiquitous that if you inhibited calcium, you know what you would do is you would kill yourself. So don't avoid calcium. Let me just go ahead and throw that out there. It is vital, not to mention your bones are going to constantly be leaking calcium if you're not going to be consuming it. So good luck. You're just going to give yourself osteoporosis and you're going to be none the better for it. It's not going to help your blood pressure at all. So that is not one that you could downstream, but EGF and TGF and Emilin, but specifically TGF and EGF are probably ones that you might want to look into if you have high blood pressure and see and you could get some of this stuff done. This would probably be really expensive, but if you could figure out if you have an Emilin deficiency or a decrease in Emilin, then that could tell you a lot of information on how you might potentially be able to treat yourself. So a lot of it's out there. It's kind of cutting edge stuff. It's not like, oh, I'm just going to fast or I'm going to drop weight, which of course all those things help, no doubt. But the mechanisms, trying to understand those can be really, really complex. Okay, so if you have high blood pressure, hopefully this was a bit informative for you. Hopefully that wasn't too overwhelming. That was literally a small percentage of the data, just as always. So there's a lot more that they ended up doing. But in terms of the overall story, that is the overall story. So don't think that they came up with a solution and I just kind of cut it out for time or brevity or anything like that. Okay, well, thanks for stopping by. I always appreciate it. And until next time, have a good one. See ya.